name is Sangeeta, and I work at San Diego State in the Brain Development Imaging Lab with some of these lovely people over here. Thanks for coming out, guys. And so what we do is a lot of different kinds of investigation of brain differences with individuals with autism. So today, as Francesca mentioned, I'll be talking a bit about typical brain development and how that might go awry in autism spectrum disorder. No. We can just make a whole new PowerPoint right now. If anybody. I don't know how that works. Oh, I think this is the little picture. From the beginning. Perfect. Sweet. All right, so that's me. Autism is a neurodevelopmental disorder. It's highly prevalent, now estimated 1 in 45 children are affected by autism spectrum disorder. And it's characterized by impairments in social communication and interaction, and by the presence of repetitive behaviors and restricted interests. And symptoms typically manifest around 2 to 3 years of age. Deficits can be summarized by 2 general domains: social communication and interaction, and repetitive behaviors and restricted interests. And in terms of the first domain, social deficits typically manifest in ways where individuals with autism might have trouble making and maintaining eye contact, might have difficulty understanding the perspective of others, and also by maintaining peer relationships and friendships with individuals. And in terms of the second domain, individuals with autism may engage in repetitive behaviors such as hand flapping or rocking back and forth, and individuals with autism may also exhibit restricted interest and insistence of sameness, sometimes becoming extremely focused on a specific object or topic. ASD is a heterogeneous disorder, which means that symptoms may definitely vary significantly across individuals. And contrary to some belief, kids with autism look vastly different across the spectrum. Any of the behaviors that I just mentioned can vary widely. For example, individuals with autism may range from having very poor language ability to having very focused and very formal and developed language. And while some may be quite aversive to some sensory stimuli, they don't like being touched, others may actually prefer like very hard pressure. If any of you are familiar with occupational therapy clinics, you might have seen the giant squeeze machine, which is just this big foam pad and a wedge that goes over, and some kids absolutely love it. They come in and they're like, I want to be in the squeeze machine. And others are like, I don't even want to see it in the room. So individuals with autism can vary quite significantly across the spectrum. It's said that if you know one kid with autism, then you know one kid with autism, and that phrase definitely speaks to the heterogeneity that you see in the disorder. Currently, and then I have some really cute figures of the kinds of social isolation that kids with autism often exhibit, routine to sameness, and then sensitivity to sensory input. Currently, autism is diagnosed by behavior and observation, and behavior intervention is one of the most successful treatment options. And in the field, there's definitely a push to develop biologically based markers that we can use as diagnostic tools. So tools that we can use when we look at either genetic evidence or brain-based evidence that we can reliably and consistently use to diagnose autism, which right now doesn't exist. So what is typical development like? The relationship between brain structure and function isn't very simple. Oftentimes, your brain is using multiple brain regions to complete any given task. Even though we hear about language centers and emotional centers of the brain, it's often a distributed network of regions that are working together to complete anything, even when you decide to reach out and pick up your beer and take a sip. That's definitely activating a distributed network of brain regions. So these multiple brain regions that activate and work together in response to any different cognitive demand is called a network. So this brain is just illustrating that for any given task, there's definitely multiple brain regions that are distributed that are activating. Physiologically, oh, and then there's here, kidding. So he's cute. So physiologically, these networks are comprised of large groups of neurons or brain cells that communicate with other groups of neurons through synapses. And these synapses connect the brain cells to each other and kind of form the wiring of the brain. So it's just an example of a very microscopic element of the brain itself. So early in development, the typical brain undergoes a process called synaptogenesis. And what this means is that during around this time, ages 2 to 3, synapses are formed at a faster rate than at any other point in development. And this produces about twice as many synaptic connections that will be retained in adulthood. So again, by age 3, we truly have about twice as many connections in our brain than we'll retain, and this allows for a wide range of possibilities that can occur during development. For example, being able to pick up any language that's being spoken in the house. So while genes arrange the basic wiring of the brain, environment and experience plays a key role in development. And this happens through a process called synaptic pruning. Pruning rids of connections that are not being used. It's in this way that the environment influences how a child's brain processes information. So by strategically eliminating connections that are not being used, resources can then be spent strengthening connections that are being frequently used. It's said that neurons that fire together wire together, and this specialization provides a developing brain with efficient functioning on a global level. So on a global scale, efficient brain functioning is characterized by two complementary principles called functional segregation and functional integration. And functional integration within a network is where different regions are communicating to complete a specific task. So you have these strongly connected, distributed brain regions that are working together to complete any task, and that's integration. There's also functional segregation between networks, where separate, specialized networks are differentiated for different tasks. And I'll go into this a little bit more in a bit. For example, tasks involving language and visuospatial abilities would involve different brain regions and therefore different brain networks that observe these tasks. And these segregated networks develop as individuals master functions that support these tasks. So as you learn more about the specific language when you're around 2 or 3 in your environment, your brain is connecting these regions that are working together for, say, English or other languages. And visuospatial tasks, which are going to be using a different series of brain regions that activate different networks. So, what do I do? I work in the brain development imaging lab, and here we use a bunch of different non-invasive imaging techniques to investigate how the brains of individuals with autism are different from typically developing individuals. So, using a magnetic resonance imaging machine, we can derive a bunch of different kinds of measurements. We can get information about the anatomy and the brain tissue, as seen by an anatomical MRI. We can get information about the white matter tracks, so basically how where the highways are in the brain, and that's seen in diffusion MRI. And we can also get functional MRI, which tells us kind of how the brain is communicating with different regions. So, for example, diffusion MRI can give us a lot of information about the structure of the brain, while fMRI will give us information about how the brain is functioning. So, what do we know about the brain development of individuals with autism? So, on a microscopic scale, some of the literature that we see supports accelerated synaptogenesis. So that process that we were talking about earlier, where in a typically developing brain, the brain is creating these synaptic connections with the rest of the brain to kind of prime the brain to be ready for any kind of environmental influence, individuals with autism tend to have more synaptogenesis. And we also see a synaptic pruning deficit, where less of these connections are being pruned away, less of these connections are being removed based on that environmental influence. And on a more global scale, we spoke about integration and segregation in typically developing individuals. And in individuals with autism, we tend to see reduced integration, where regions A, B, and C within a network are communicating in a less efficient and less robust way than individuals in the TD brain. And you can see that by the thickness of the line in this diagram, which is completely hypothetical. There are no ABC regions we know of. So we can kind of picture the reduced integration within these neurotypical networks by this diagram. And we also see reduced segregation between relevant networks, where regions A, B, and C within a network are also connected with out-of-network regions D, E, and F. Here in this diagram, we can see that weaker communication within neurotypical networks and more residual and excess communication with brain regions outside of neurotypical networks, this can introduce unnecessary noise into the system by both accelerated synaptogenesis and a decrease in synaptic pruning. We can see this kind of communication with regions that are outside of neurotypical networks. And this impact of noisy processing and reduced specialization in the autistic brain may be demonstrated by this example of daily perception and behavior that I found on Facebook. But it was interesting. It was this autism poster that was recently published or presented from our group, and there were a lot of comments. And I like this one because it really demonstrates how this noisy processing is actually translating into sensory perception of noise. The first user says that some of the time in highly emotionally or physically stressful situations, I have to close my eyes to hear things. And a user responded that he can't process more than one sense at a time. Sometimes he needs to cover his ears in order to be able to think. So we're really looking at these systems that typically are segregated, that are kind of overlapping and producing too much actual literal noise in this case. So just as a last point, it's unfortunate that this is still a talking point, but there's actually no evidence that autism and vaccines are linked. The actual initial study was completely retracted. All of the co-authors actually removed authorship from that paper. Andrew Wakefield, I think, lost his license. And in 1998, the next year, there were a plethora of studies that have come out since 1998 that show no relationship between vaccines and autism. This exact study was actually not a controlled experimental study. There was no control group. It was very, very biased, and later investigations actually reported that Dr. Wakefield actually falsified a lot of the data. So on so many levels, it's not a real study. And everything that's come out since points to no relationship between vaccines and autism. But we do notice that, I would like to point out that the timeline of when children normally get vaccinated definitely overlaps with the timeline that we see the onset of autism symptoms. So when we hear those anecdotal stories about the kid that went and got vaccinated, and then we see these social symptoms and repetitive behaviors that are manifesting at a similar time, there's definitely some confounding issues there regarding the timeline where all this stuff is happening. It's happening at the same developmental period, which really, again, emphasizes and highlights the importance of controlled experimental studies where we can observe these different things in a scientific way. And just in case that didn't convince you, the actual ingredient that was said to be one of the causes of autism, the marisol, was removed from vaccines in the year around 1992, and the prevalence of autism still rose, which should show you that the ingredient that was thought to be related to autism is not related to the increasing prevalence at all. Which kind of brings me to my next point of why this increasing prevalence of autism. And this could be for a number of reasons. One of which being that the diagnostic criteria by which we view autism and by which clinicians actually diagnose the disorder has changed to be more inclusive, to include kind of, when I talked about the wide range of symptoms and heterogeneity of the disorder, Recent diagnostic tools have changed to include such a wide range of people. Instead of looking at the very narrow, the narrow group of people that are very low functioning, it has changed to include more of the higher functioning individuals that might have some social difficulties, repetitive behaviors that might not be impacting daily function severely. And so if there's a consensus about, that autism is brain-based, why do we still diagnose based on observation and behavioral criteria? And the answer to this is really what my scientist friends and I, and the community is really trying to figure out. We definitely need biomarkers that I mentioned earlier. We need some sort of reliable, consistent way to diagnose the disorder, which might actually be related to the identification of brain-based subtypes. If we can identify different subtypes based on brain patterns, we might be able to explain more of the variance in the kind of samples that we see that range from individuals that are nonverbal to individuals that are super chatty, and if we can kind of identify more of the variability, then we may be able to develop some biomarkers. And measures of network connectivity using structural and functional MRI and a bunch of other amazing techniques, we hope to produce such biomarkers. My thank you slide didn't show up very nicely, but thank you. Any questions? Sure.
Is the rate of autism the same in third world countries versus first world countries?
So that's a really good question. He asked if the rate of autism was the same in third versus first world countries. And the prevalence rate does have a lot to do with, I mentioned the actual diagnostic criteria, but also a lot of the time in first world countries, we have individuals or families who are really trying to get services for their child. That has to do with an official diagnosis. So we do see the prevalence a little bit higher in not only first world countries, but places with higher socioeconomic status. Families that might have a little bit more resources to devote to behavior intervention tend to, or school districts that might provide better services, there might be more of a push in the community to actually get a formal diagnosis, be able to obtain these treatment options for their kids. So especially in some third world countries, for example, India, one of the people that I'll be working with shortly is actually working closely with people in India to try and not only increase kind of the knowledge base about autism in the area, but what kind of treatment options there are. And with kind of the information of diagnostic criteria, we're getting more education kind of all around the board, and with that comes more increased rates of diagnosis. Does that make sense?
So I work next to a lab that's focused on using MBPA to study brain stuff in general, and I myself have very little understanding how the brain works, but has there been any attempts to run some machine learning classification techniques on how we can predict some of that autism based on, say, neural factors or behavioral factors or some combination of those?
So I'm not sure about MBPA exactly. I haven't read too many studies about MBPA exactly, but when I mentioned kind of all of these different measurements of network connectivity and the way that the brain is functioning, we have structure, we have methods that can show us very great resolution in time, very good resolution in space. But all of these different methods kind of speak to different strengths and weaknesses of the method itself. So there are definitely, I think the field is kind of moving towards using large machine learning, algorithmic-based approaches to kind of make sense of all these data. The minute we start including like genetic sequencing and genome-wide, we're really just throwing, like there's a lot of information to work with that all speak to different aspects of what could be going awry in autism. So I think the field is definitely moving towards, yeah, is that what you said?
Yeah.
Yeah.
Are there any good animal models that show both biological validity as well as space validity?
Sure. So I don't work particularly with the animal research, but from what I know about the strengths and weaknesses, and correct me if I'm wrong, anybody else that's here. But from what I know about the animal models is that though, for example, I think one particular study looked at something called the SHANK3 gene, and by actually simulating an environment where a mouse can, or a rat can kind of choose between either a cage that might have another rat inside, some kind of a more social decision, or just like food. So maybe food is in both. Or those kinds of simulations definitely do point to specific abnormalities. But the issue here being that nothing, even in the imaging literature, in the human models and the rat models, nothing's coming up as quite consistent and reliable as a biomarker. And if you think about animal models and sociability, it's a little bit harder to model, but you definitely do see rats that have more of a preference for being alone with the SHANK3 deletion, for example, which is does that? Okay.
So I know that we don't know like what causes autism, but if I was to get an autism researcher alone in a small room with some friends over a beer, and the camera's not on you anymore, would I be able to get her to speculate on any suggestive possibilities that?
So I don't think I have the best answer for this. I don't think so. I don't think that we really know what could be the cause. I think because, and I really want to highlight kind of the heterogeneity of symptoms again. You know, it seems like a really nice thing to keep bringing in. When we don't know an answer, we'll just talk about the heterogeneity. But really, that heterogeneity of the symptoms, by kind of isolating brain-based subtypes, we might be more likely to actually come to different causes. For example, if anybody knows that girls with autism are quite more rare. I think it's like 1 in 4 or 1 in 5 girls with autism has a diagnosis. So that, so even just that kind of gender variability might speak to different causes. I don't know if that makes sense. By seeing such different patterns of symptoms and behavior, I don't think that one kind of really nothing's going to wrap it up real nicely with a bow. I think that we have a lot of different kinds of mechanisms at play here. That's kind of what's making it difficult from the research end to kind of parse apart what